Hi, my name is Elaine from the Charter School Wilmington, and my research topic from the technology field, specifically biotechnology, is genetic modification a valuable bioengineering tool or an unethical way to change our natural world? A new world is unfolding, one where modified plants and animals are increasingly common, genetic diseases are cured, and even extinct species may roam again. The application of gene editing technologies make this possible. As the technology progresses, the possibilities of gene modification seem endless. However, people continue to question whether these benefits come at the cost of ethics, morality, and social fairness. Genetic modification is not new. With the definition of genetic modification as any process that manipulates an organism's gene pool, people have been developing this technology for centuries. Humans have been using it to manipulate genetic traits for millennia through selective breeding or artificial selection, cultivating sturdier crops and breeding animals with desirable features long before scientists understood heredity. In the 20th century, mutagenesis enabled the creation of desirable traits in new species. Researchers used ultraviolet radiation and ethyl methane sulfonate to accelerate mutations and improve crop varieties. A real breakthrough occurred in 2012 when CRISPR-Cas9 demonstrated the power of precise DNA modification. CRISPR-Cas9 is an enzyme system naturally found in the immune systems of bacteria. CRISPR recognizes viral DNA and Cas9 destroys it. Scientists repurpose this system to cut DNA at specific locations, allowing genes to be added, removed, or altered with unprecedented precision. Genetic modification involving DNA editing within our food system provided the first controversy. GMOs have transformed agriculture by increasing crop resistance to pests and diseases, leading to higher yields and less pesticide use. Bt crops contain genes that produce insecticidal proteins, improving yields while reducing pesticide use. On a global scale, GMO crops have provided more affordable food and reduced global food insecurity. Due to consistently high yields, GMO farmers realize higher incomes, leading to increased financial security. Reduced tilling minimizes soil erosion and improves carbon retention, contributing to sustainable agriculture. In this graph here on the right, the use of GMOs in corn in Nebraska has led to a significant economic increase over the span of 20 years from 2001 to 2021. Corn genetically modified with both Bt, Bacillus thuringiensis, and HT, herbicide tolerant, are used for pest and weed management. As the percentage of GMO corn increases, so does the money in billions of dollars. Despite these benefits, serious concerns remain. Herbicide resistant crops are associated with the loss of monarch butterfly habitats due to the destruction of milkweed. Superweeds continue to evolve resistance, triggering stronger pesticide use. Some GMOs contain antibiotic resistance markers that could transfer to human gut bacteria. Here, the picture shows petition surfacing of a GMO free USA as a result of pesticides, habitat loss, and extreme weather on the monarch butterfly species, making them endangered. Public acceptance of genetic modification exists at different levels. People easily forgive unconscious genetic modification, such as selective breeding, because it is viewed as part of humanity's survival experience. Many view selective breeding as an extension of evolution, where humans accelerate natural selection. Large populations also embrace plant hybridization since both parents are natural species. However, intentional DNA editing has sparked heated debate. Judging genetic modification by how artificial it appears rather than the actual effect it brings results in an inconsistent evaluation process. When a gene from a Brazil nut was introduced into soybeans, nut allergic consumers reacted adversely. This example illustrates how genetic alteration can unintentionally introduce new health risks, emphasizing the need for careful testing and traceability. The invention of CRISPR-Cas9 put human gene editing at our fingertips. CRISPR enables the potential treatment of over 8,000 monogenic diseases, including sickle cell anemia and cystic fibrosis. Instead of a lifelong treatment, gene editing offers the possibility of permanent cures. DNA editing raises serious safety and ethical concerns. A study led by Sushita showed that CRISPR editing caused chromosome deletion in 5 to 20% of cells. Beyond safety, enhancement raises fear of social inequality, while genome design in embryos threatens to alter future generations. Genetic modification is never an isolated decision. Structural biologists, bioinformaticians, bioengineers, and clinical researchers all contribute to extensive testing and evaluation. New approaches like epigenome editing allow reversible changes. With regulation and ethical oversight, genetic modification is not unethical but essential for society's greater good. Thank you, and here are my sources. The sources continue on this slide. Thank you for listening.